think next we have Professor Katherine Villa from ISIC, the Institute of Chemical Research of Catalonia. So Katherine obtained her PhD in chemistry in the Autonomous University of Barcelona, and now she's a group leader at ISIC. And she has, also received many accolades and awards for her research work, including the Young Research Award by the Royal Spanish Society of Chemistry, the Leonardo BBVA Award, La Caixa Junior Leader, and she's also an elected member of the Young Academy of Spain, right? And more recently, I think it was last year or a couple of years ago, she got an ERC starting grant, which is like one of the most prestigious awards that you can get as an early researcher. So I think these are also a good switch of topics, right? Because she's gonna talk about using light instead of chemistry and biochemistry to power microrobots. So thank you very much, Katherine.
Thank you. So good afternoon, everyone. Thank you for the kind introduction, and also I would like to thank the organizers for the invitation. So my talk is about the design of micromotors that can be activated and controlled by light. And our inspiration comes from nature, like for instance, if it works. No pasa. En la pantalla. No. Well, I tried before and it was working. So. No.
No has enchufado nada, ¿no?
No.
¿Te parece si salimos de pantalla compartida?
Vale. Claro, pero ahora... Pero vuelvo a donde estaba, ¿no?
Ya, yo creo. A ver si te deja. ¿Echamos? Prueba. ¿No? ¿Y por qué?
¿O le conecto esto?
No, es que está petado el ordenador.
No, porque sí que avanza.
Vale. A ver. Un segundo. It had to happen. Almost the last presentation. Okay. A ver. Ahí. Es que le das a proyectar y no tira.
Ahora.
Ahora.
Vale. Sí.
Okay.
Okay, thank you. Sorry again. I have tried before and it was working, but. So our inspiration comes from nature. From nature, for instance, photosynthetic organisms like cyanobacteria demonstrate how light is a powerful source to drive motion and also induce some biochemical processes. So inspired by this, we can create micromotors that contain photoactive components. And in this way, we can create micromotors that move and adapt to the light stimuli, but also that can sense and interact with their surroundings. So some of these biomimetic features include, for instance, their capabilities to perform a 3D alignment when we shine the light, for instance, on the top. So on the left, here we can see some algae that have this characteristic that once the light is on top, they will self-align. And then on the right, some artificial nanomotors with a rod-like morphology that will self-align according to the light beam. Also, since they are photoresponsive, so we can trigger their motion and control it by using light irradiation. Again, on the left, we can see some photosynthetic organisms, and on the right are micromotors, how they move upon light irradiation. But also, the aquatic organisms have this capability that they can sense where is the light source, and then all of them can go and accumulate there to have higher nutrients and obtain their own food. So in a similar way, we can create micromotors that also signal others, and then they will accumulate where is the maximum light beam. So how these micromotors work? Well, it is a light-induced chemical process. Since the micromotors that we develop, they have photocatalytic materials. Once we shine the light, this material absorbs the light. It will generate electrons and hole pairs that will react with the species absorbed on the surface, and then it will perform some energy conversion that finally will lead to the motion, but the asymmetrically release of this chemical species. So the micromotors move by phoretic mechanisms, but also, as we can see here, the releasement of these protons or hydroxy also are the responsible for these micromotors to interact with the other particles around or with other entities. So while light is a powerful source to induce their motion, also there are still some remaining challenges regarding the design of these photoactive micromotors. For instance, one of them is the fabrication methods that are actually very expensive. Also, these micromotors usually require platinum or gold in their structure for be efficient, which also increases the cost. But also, the main problem is that after the fabrication process, the amount of micromotor that we obtain is very little. So the yield is very low. So for instance, if we want to use these micromotors for an environmental application, we want to have like tons of these micromotors. Another limitation is the optical properties. That is what tells us which wavelength we can use to activate them. And unfortunately, most of them are more efficient in the UV range. That only corresponds to 5% of the solar spectrum. So that's why we want to develop micromotors that are able to move under visible light. And also, due to their tiny sizes, there are also challenges regarding the material design, how we can fabricate or combine different types of materials on the same structure and then have different functionalities and properties. So to overcome some of these challenges, in our research group, we are working on the development of micromotors that have anisotropic morphologies. Also, they are based on only single component materials, like semiconductors and organic semiconductors. And also, we fabricate them by upscalable methods, like sol gel, hydrothermal, co-precipitation, and usually in one-stop synthesis. And also, most of them are activated under visible light. So in our research group, we have been working on studying different aspects of these photoactive micromotors. For instance, how we can manipulate light to induce collective interactions among these micromotors. Also, how we can provide them with advanced functionalities that they cannot only move, but also sense the temperature of the media. And recently, as Abdon said, thanks to our ERC project, we are developing a novel class of photoactive micromotors that are photorechargeable. So the idea is that they can absorb the light, store it, and release it when needed in environments where the light cannot penetrate enough or the samples are highly scattering media. And also, the main application that we have been trying this type of micromotors is for environmental remediation, most specifically for water purification. So starting with the first one. So one unique feature that these micromotors have is that as soon as we shine the line, all of them will self-assemble. It is on the right field, and then on the right is in the luminescence field, since they are fluorescent. So here we are just irradiating the whole area. But if instead irradiating the whole area, we selectively irradiate only one single spot, we can see how they will self-assemble and accumulate there, but also will follow the light pattern. If we increase the size of this light beam, then they will self-assemble in larger clusters and will also follow the pattern. And if instead of increasing the size of the beam, we increase the size of the particles, then we can have like a more dynamic self-assembly formation. And since everything is induced by light, we can control this self-assembly, disassembly by turning the light off on, as we can see here. But also it is reversible. We can achieve it as many times as we want by just switching the light on-off, as we can see here in the second video. So the next step was to see if we obtain this formation of these self-assembly, like in 2D, or if they are really agglomerated in a three-dimensional way. So we perform some Z scan imaging. And actually, we corroborate that indeed it's like a 3D self-assembly of these micromotors once we shine the light. And also, we can not only obtain these like circular shapes, but we can even achieve a more complex self-assembly by modulating the light pattern. So they can follow, like if we use a rectangular shape, they will adapt and will form this self-assembly. Here we have like multiple circles, and they will also form these different self-assemblies, or even more complex, like spirals, or even heart-shaped morphologies. So what's interesting here is that depending on the pattern that we use and the morphology of the light irradiation, we actually can get different heights of this self-assembly. So for instance, taking as example these multiple circles, we have performed some 3D mapping of this data scan, and the circles that are very tiny, as you can see, the height is not very large. However, the ones that are in the outer part, they can reach like larger pillars. And actually, as we corroborated here by doing this 3D set scan, most of them will agglomerate in the middle and then form the larger clustering on the top. And actually, when we just shine the light, they will form like this very ordered self-assembly, as we can see here. But after long-term irradiation, they will destroy this nice self-assembly because they are coming one on top of each other. So the takeaway message of this part is just that by light manipulation, we can actually induce very sophisticated and collective functionalities in these photoactive micromotors. But besides this self-assembly, we can develop photoactive micromotors that can perform temperature sensing. So in this case, what we did was to combine two different optical materials in the same structure of the micromotor. So we have the zinc oxide that is necessary for the motion and the light irradiation, and then we have the upconversion particle that is able to, under near-infrared light, to give us the optical signal for the temperature measurement. So this is just a two-photon process that I will not explain in so much detail, but it's just these codopants of the upconversion are thermally coupled. So depending on the temperature of the media, the ratio and the intensity will vary. So the micromotor that we develop can sense, for instance, in this case, from 297 to 330 Kelvin. And here we can see how they can self-propel under light irradiation, and on the right, how by switching the light wavelength to near-infrared, they have this green emission that is not only useful for this temperature sensing, but also for tracking purposes. So this dual functionality allows us to have micromotors that not only move, but can also monitor their environments. And in this last part, what I will explain is how we can apply these micromotors for water purification. So what we do is that we put these micromotors in water, and then we irradiate them with a solar simulator. So we use them, for instance, for capture heavy metals or remove organic pollutants, and also even for inactivation of pathogenic microorganisms. So here we developed the first example of a totally metal-free tubular micromotor that is able to move under visible light. This micromotor is made of carbon nitride, and also we didn't use any template or other complex methods during the fabrication. It's just a supramolecular self-assembly. And as we corroborated by SEM images, they are made of only carbon and nitrogen, so in principle they are biocompatible. And also they exhibit this intrinsic blue fluorescence. So once we shine the light, the micromotor will start moving. This type of micromotors require hydrogen peroxide as chemical fuel. Once we stop the light, the micromotor stops the motion, and it can be easily reactivated by shining the light again. And since they are fluorescent, we can also follow them by fluorescence microscopy. So the next step was to evaluate what happened to these micromotors when these different heavy metals are present in the water. And what we observed was that in the presence of these different heavy metals, the micromotors actually move much faster at the same time that they are absorbing and removing these heavy metals from the water. So for instance, if we take as an example the copper, the micromotor move much faster, but also in this case, then they require less concentration of this chemical fuel. So from an environmental point of view, this is very promising because then we can harvest the pollutants that are in the water for the micromotor to be more efficient, but at the same time to remove this type of contaminants. And the explanation for this is that once they are in contact with these heavy metals, they will bind to the surface of the micromotors by electrostatic attraction, but also by acid-base Lewis interactions. And when these metals are adsorbed on the surface of the micromotor, these metals, like the copper, will contribute to decompose the chemical fuel. And that's why the micromotor moves faster. So as a proof of concept, we use them to remove these copper ions. Here we have a comparison of the performance of the micromotors under the classical magnetical steering. We could achieve almost 35% removal in 7 minutes. If the micromotors were not moving, they achieved almost 40%, but in a longer time. However, when the micromotors were actively moving under light irradiation, we achieved almost 55% in only 7 minutes. And if we use a 1 ppm solution of the copper ions, they could achieve a 100% removal. So this demonstrates how their active motion and the generation of these bubbles that enhances the micromixing can contribute to have higher removal yields. And since they are fluorescent, we can also have like an insight of this removal process. Here is on the left is before adding the copper ions, and after that, the fluorescence is quenched. And actually, it is related to the different concentration of these copper ions in the media. So these results demonstrate how we can create micromotors that are very low cost and are very efficient for addressing this type of contaminants in polluted water. But in this case, the micromotors are just absorbing the pollutant, so they are not really like degrading it. We need to remove the micromotors to take the pollutant from the water. So what we did next is also to develop micromotors that can not only absorb the pollutant, but also degrade them as, in situ while we are irradiating. So we have the fabricated micromotors that can degrade different types of pollutants from the pharmaceutical, dyes industries, and so on. But what I'm going to show here is a further step on how we can also, by modifying the surface of these micromotors, we can achieve like a more selective capture of a specific pollutant. So instead of we just put it there and they will degrade everything, we can modify the surface to achieve like a selective pollutant removal. And this is very important because usually real wastewater samples contain more than one pollutant in the same mixture. And once this water is treated, the ones that are the most toxic ones will remain, that are highlighted here in yellow. So what we did was to modify the surface of our micromotors to target these specific pollutants that are very toxic, and in this way, to increase the degradation yields. So for this work, we developed bismuth vanadate micromotors that are also active under visible light irradiation. Here we use just a hydrothermal reaction, and we can see that all of them looks quite similar. They have a butterfly-shaped morphology. So in this study, what we used was paranitrophenol, which is a very toxic pollutant from the chemical industry. And then we polymerized the surface by surface molecular imprinting. And in this way, we can create like a mold on the surface of the micromotor with this pollutant as template. And once we remove the template, we can get surface recognition cavities on the surface. So it's like hands that will take this specific pollutant. So the next step was to see if indeed we achieve this polymerization process, if we achieve the formation of these surface cavities. And as we can see here, on the left is the unmodified one, and then after the polymerization process, we can observe the formation of this carbon layer, as we also corroborated by EDX mapping. And also, since they are photoactive, the other concern was if the polymer is covering the whole surface, if they will still move. But as we corroborated here, the presence of this polymeric layer on the micromotors didn't hamper their motion. So they are still able to move under visible light irradiation. So once we achieved on obtaining this, we evaluated their performance. And actually, as we observe in this plot, the modified ones achieve a higher absorption of this paranitrophenol. And in this way, we can increase the decontamination of this pollutant from 27 to almost 50% in only 30 minutes. That if we compare it with the literature, this gives us like the highest removal rate, a very short time, but also using visible light, which is more challenging. So the final step was to prove our concept, and then for that reason, we combined this paranitrophenol with another pollutant, in this case, methylene blue, and then we evaluated if the micromotors were indeed selective towards one single pollutant. And as we can see here, we could increase from almost 15% from the unmodified motors to the modified one to almost 60%. So this is a huge improvement, and this demonstrates that surface molecular imprinting is a very promising approach for providing these micromotors with this selectivity, especially when they are based on the generation of reactive oxygen species that are usually non-selective. So just to finish this part, I will show also how we have fabricated micromotors for the removal of pathogenic microorganisms like bacteria and also fungal cells. So here we can see like a macrophage, how it autonomously go and capture bacteria and kill it and eliminate it. So in a similar way, we have developed visible light-driven micromotors based also on bismuth vanadate that can move and autonomously go and grab these yeast cells in this case. So here is the video. The black particle is the micromotor moving under light irradiation. It rotates and it goes and grabs the yeast. If we turn off the light, it will release it. And it doesn't matter how far the micromotor was, it still goes and swims towards the yeast and capture it. And it can even capture and transport big agglomerates of cells. So in this study, we didn't use any magnetic guidance or any surface modification. It's like an intrinsic property that these micromotors showed in the presence of these microorganisms. And also, we evaluate if the micromotors were still also able to go and grab these yeast cells in the presence of other microorganisms, like E. coli. And as we can see here, that star is the micromotor, and I don't know if you can see it well, but there is a E. coli attached to one tip of the micromotor, but it still goes and swims towards the yeast cell. So taking advantage of that the micromotors release reactive oxygen species and this enhanced contact, we use them for the inactivation of this fungal contamination. Here we have some microscope optical images. The green images correspond to the live cells and the red ones to the dead ones. And we can corroborate that after the treatment with our micromotors, almost all the yeast cells are dead. And even their contact is so strong that even after the process, the yeast cells remained attached to the surface of the micromotors. So this is one example of how these micromotors can remove this fungal contamination, but also in order to reduce the environmental footprint of these micromotors, we have also developed self-degradable photoactive micromotors that we shine the light, they will kill the bacteria, and they will disappear from the media. So for this purpose, we fabricated micromotors based on silver phosphate with a tetrapod-like morphology, as we can see here. Once again, these micromotors are active under visible light, and also all of them look quite homogeneous. We fabricate them by co-precipitation method. And they can move in water and also in the presence of biocompatible fuels like glucose, everything controlled by light. And then since this silver phosphate is photosensitive, what happens is that over long irradiation periods, it will start to release these silver nanoparticles from the surface. And it has like this etching. And we analyzed the particle size of these silver nanoparticles that they were releasing, and that it's about 500 nanometers. And this is just screenshots of how the micromotors under long-term irradiation, they start breaking apart and disassembling. And this is due to a self-reduction process under light irradiation. It will generate these electrons and the silver ions, and then it will reduce these silver ions into silver nanoparticles. But what was surprising is that when we combine these micromotors with bacteria, like Staphylococcus aureus and also E. coli, we observed that this self-degradation process became much, much faster. So in only 1 minute, the micromotor was totally destroyed after this 1 minute of light irradiation. But as you can see, once they are moving around, they are releasing these silver nanoparticles as well. So once these nanoparticles are released in the media, they can interact with the bacteria and then, of course, will kill it. But to get an insight about this, a more quantitative insight about this antimicrobial activity, we perform different blanks. Like, for instance, when we have the micromotors that they are attached to a substrate that they cannot move, we can see that the removal of this bacteria is almost, is very low, it's around -1. However, under light irradiation, the performance is a bit bigger. If we add directly silver nanoparticles that are very well known to have a high antimicrobial activity, we see some improvement, but there is no any difference under dark and light irradiation. However, when we have our micromotors actively moving under visible light irradiation, we could achieve a 99.999% bacterial removal. And this is attributed not only to their capabilities to move around to generate silver nanoparticles, but also to the release of these reactive oxygen species. In particular, we found that the superoxide radical is the main responsible for the killing of the bacteria. And we can also use them for inactivation of biofilm that are much resistant than the planktonic cells. And once again, we have higher performance of these micromotors under light irradiation. So by combining all these 3 factors, releasement of ROS, self-propulsion, and releasement of silver nanoparticles, we can have really, really effective antimicrobial micromotors. So just to sum up, I have shown how we have been using these micromotors for different types of applications in the environmental field, but also how by light shaping and material design, we can also provide these micromotors with new functionalities and also with sensing capabilities. So before finishing, I just want to thank also my research group, our funding sources, our collaborators, and of course, to all of you for your attention. Thank you.
Thank you very much, Katherine. Do you have questions from the audience? Yeah, please, Nils. And I think for the people online, it will help. Yeah. Thank you.
Thank you for a super interesting talk. So my question might be unanswerable, but let's see. So what effect do these motors exert on the surroundings? Because some of the, I mean, I kind of get like ant visions, because they seem to be carrying around at least stuff way bigger than themselves. So do you know about the effect they can actually exert, like in wattage or horsepower or whatever measure you could think of?
Well, no, we haven't measured, but they provoke a fluid motion as well as they are swimming in the surroundings. But at least me, I haven't measured, but I think that other groups have performed it for other type of micromotors, no?
We have measured in the past the micro and nanomotors that they show, and they are the nano were at 60 femtonewton average, and the micron were 14 piconewton. So it's the same level of a molecular machine, which is almost very, very small, right? I mean, but we're not able. Probably that would be a challenge, right? With optical tweezers, you cannot do like any metallic material because like absorbs the light, and we'll see how it destroyed. But there might be other ways to measure. Maybe they will be not at the piconewton, maybe larger, but because hair particles were moving larger, so
No, but
micro.
with an end effector, I think you can do that.
Yeah.
Yes,
But
micro cantilever.
Yeah.
So, yes.
Yeah, thank you very much for a nice presentation.
Thank you.
Yeah, this may seem low, but it's like it's in the range of you can manipulate cells for this, right? And there's a lot of this technology being used for like in vitro fertilization, where you can manipulate the eggs and everything, right? So it's again the right tool for the right job, right? Yeah. Exactly. Yeah.
Yes.
Thank you.
Thank you, Katherine, for the next talk. So I was thinking about, is there any way to recapture all the microbots? Like once you drop them to get the different things, right? Imagine that you want to capture something to analyze later.
Yes, usually by modifying the surface with the magnetic nanoparticles,
Okay.
then we apply the magnetic field, and then we can recover them. Yes.
Okay,
Thank you.
thanks.
Thank you. I do have a question. If there's another question, it's hard to see, but what's the mechanism for the light-mediated self-assembly? Like, what's the actual particle-to-particle interactions that makes them cluster?
Well, this happens in the presence of a redox shuttle that is benzoquinone-hydroquinone. So once we radiate it, they are generating protons and hydroxy groups.
Got
This
it.
is causing this
Okay,
interactions.
I missed that. Yeah, sorry.
Yeah.
Yeah, thank you. Well, please let's thank Katherine.